Okay, so first of all, I am deeply sorry, very, very, very sorry, but I have to make an announcement. I will not be here. You can go out, yeah, don't worry about it. There is a conference at the end of July, August, so we have to make up lectures and we have to reschedule the exam. So the advice I had from Jimu is, no, it's not so bad maybe, but the problem is that it's on Saturday. So on Saturday the 17th and Saturday the 24th, we are having, you also know this one. So we will be 3 hours. We will be more than 3 hours, 4 hours and a half. So 17th and 24th at the 1, 2, 3, 4th period, we are going to have lecture on the 17th and the 24th. Okay, I am not sure yet what I'm going to do, but since it's on Saturday, I kind of may want to do a little bit something different from normal. Well, it's not normal lecture that we are doing, but Nanka, I will think of something. Alright. Okay, so if you cannot make it, it's probably okay, but I will be there, so if you want to come, it will be nice. And the test, as you know, this doesn't change, so the midterm exam is on the 15th. Midterm exam is on the 15th, and the final exam will be then on the 20th of July. So it's, I think, 1 week earlier than normal. So you got this, okay? And I'm sorry. All right, okay. So we have almost finished this review paper on the transcriptional architecture of the mammalian circuit clock. So let's just continue reading. This time is your homework.
Given the circadian rhythms of RNA polymerase II, chromatin states associated with RNA polymerase II transcription have been examined during the circadian cycle. Figure 5 illustrates genome-level views of histone modifications that are characteristic of promoter, enhancer, and elongation states. Histone H3, K4, E3, H3, K9AC, and H3, K27 AC modifications are enriched at promoters and undergo circadian rhythms in histone modifications at HER1. On a genome level, circadian rhythms in RNA polymerase II occupancy, as well as histone H3K4 and E3, H3K9AC, and H3K27AC modifications are present in the majority of expressed genes, irrespective of whether RNA cycling occurs. Both RNA polymerase II occupancy and H3K4 and E3 marks are highly dynamic and show circadian oscillations that peak during the early night for all expressed genes, cycling on RNA genes and non-cycling genes.
Okay, thank you very much. So this is just repeating what has been said before. There are oscillations of everything, oscillations in the occupancy of RNA polymerase II and chromatin state. So the whole transcriptional machinery seems to be regulated rhythmically by the clock. So of course you may wonder what regulates which. Is it the clock that regulates transcription, or is it transcription that regulates the clock? It's a cycle, so it's difficult to put the egg before the chicken. So, is there any special thing here that you would like to have more details on? No? Okay, then we are going to continue in summary, and that will be in Vietnam.
In summary, in the Maas River, there is a highly stereotyped, highly dependent pattern of core transcription factor binding, RNA polymerase II occupancy, transcription, and chromatin states. There are 6 distinctive phases of the circadian cycle. One is a void state at the beginning of the cycle as CT0, during which clock demand 1 and prime 1 want to leave outside in a transcriptionally silent state. Second is a depression phase at CT4 at the level of prime 1 prime. Third one is a major activation phase at CT8, during which clock demand occupancy peaks along with P300 and CTD recruitment and are accompanied by histone H3K4 methylation 1 and H3K9 activation marks. Four is a temporarily coordinated transcriptional activation phase at CT12 to CT15, during which RNA polymerase II recruitment and free mRNA transfer expression peak, followed by histone H3K4 methylation and H3K27 activation marks. 5 is a passage repressive phase at CT15 to CT18, during which PER1, PER2, and PRI2 occupancy peaks and transcription declines. And 6, a transition between the end of repression and the beginning of the next cycle. Interestingly, the co-activator CTP interacts with the repressor PER2 at the end of the repression phase, and CTP occupancy has a second peak at this time, perhaps maintaining an open chromatin state during the transition from repressor to activation. PRI1 occupancy also bridges this transition period and interacts with clock demand once beginning the next cycle.
Okay, thank you very much. Okay, so this is very nicely the explanation of the figure 6 of all these occupancy, sequential occupancy of these regulators of gene expression. Okay, any questions about this one? English or biological or anything is okay. Okay, then we are going to continue the next paragraph, please.
In contrast to embryonic stem cells, for which 30% of genes are paused and carry bivalent histone marks, genes that are not expressed in the mouse liver are not enriched for paused RNA polymerase II. Interestingly, the histone H3K4ME3 promoter mark lags RNA polymerase II occupancy by approximately 1 hour in cyclic genes, and the H3K36ME3 elongation mark lags by approximately 3 hours, consistent with the idea that these marks arise as a consequence of RNA polymerase II transcription. Thus, circadian transcriptional regulators are clearly involved in RNA polymerase II recruitment and initiation, as are the histone modifications associated with these events. However, additional steps such as promoter proximal pausing, pause, release, and productive elongation probably have additional regulatory roles. At enhancer sites, Clock BMAL1 is a pioneer transcription factor and can bind nucleosomes to promote rhythmic chromatin remodeling and incorporation of the histone variant H2AZ. Cycling enhancers also display circadian rhythms in eRNA expression in non-conjunction with tissue-specific transcription factors.
Okay, thank you very much. What about this one? Well, that really is a description of figure 7 and what are the different levels of control. The main one really is the initial binding of clock and BMAL and the recruitment of the RNA polymerase II and promoters. But as they say, there may also be additional controls as the pause and release of the polymerase. So it's an extremely complex thing, and not everything is understood, but that is what's understood at the current stage. Is that okay? So I guess one of the maybe main notion in this paragraph is the fact that clock and BMAL is described as a pioneer transcription factor, that when it binds, then it regulates many things that have impact on the chromatin state and the recruitment of the transcription machinery per se. So that's what is understood in saying it's pioneer. So they are the first to come and bind to EBOX. Okay. If there are no questions, then we continue on.
In the case of cycling genes, RNA polymerase II recruitment, initiation, and productive elongation are all rhythmic and occur in phase, leading to associated rhythms and histone modifications. In the case of expressed but non-cycling genes, RNA polymerase II recruitment and initiation are also rhythmic, leading to rhythmic histone modifications at the promoter. However, subsequent steps in pausing, pause release, and productive elongation may no longer be under circadian regulation. Because RNA polymerase II pausing at promoter proximal regions and pause release are major regulatory steps in transcription, it will be important in the future to determine whether pausing factors such as negative elongation factor, NELF, and DRB sensitivity inducing factor, DSIF, as well as the positive transcription elongation factor B complex are under circadian control.
Okay, thank you very much. So that's again more of the same. More of the same. Why is all this necessary for the circadian clock? Still, it's not understood. What is the physiological function of this is not really understood. Okay, and I will read the final conclusion. The circadian modulation of transcription occurs on a genome-wide scale far greater than that seen previously by gene expression profiling. The circadian clock in the liver regulates RNA polymerase II occupancy on a genome-wide basis and drives the genome-wide circadian modulation of chromatin state. These circadian rhythms in the chromatin state poise the genome for transcription on a daily basis to coincide with the daily metabolic demands of the organisms and to optimize the energy utilization of gene expression. In addition to the circadian modulation of chromatin states, the 3D architecture of the nucleus and the interaction of active and repressive chromosomal domains undergo circadian oscillations. Thus, the 4-dimensional regulation of the genome and its nuclear architecture are ripe for study. In addition to the pervasive circadian regulation of metabolism, strong links between the circadian gene network and cell growth and cancer, immune function and signaling are apparent, and components of this network will be important therapeutic targets in the future. Okay. What about chromosomal domain? So this is associated to the box 3, I think. Chromosome organization. This is the box 3, so it says, it is well established that chromatin interactions occur in 3 dimensions. However, temporal changes in 3D genomic interactions, or in both space and time, the 4th dimension, are an emerging area. The 4-day nucleosome, reference 193. Circadian cycles in chromosome organizations have been observed using circular chromosome conformation capture method at the DBP locus, a well-known closed BMAL1 target. Although chromosome interactions surrounding the DBP locus were not reported to change during the circadian cycle, the frequency of long-range interchromosomal interactions did vary. Interchromosomal interactions were highest at the peak of DBP expression and were lowest at the opposite phase, when DBP expression was low. Okay. What does it mean? Any idea of what does it mean? Of what this means? Of what this does mean?
Is it referring to like the someone else's chromosome structure? Is that basically how like the chromatin functions are?
It is not, well, partly relative to euchromatin and heterochromatin difference, but it's more than that. It's more than that.
Circadian rhythm also affect the chromosome conformation.
Yes, but in relation to that, what do you mean conformation? Especially chromosomal domain. And this is a level that is hardly spoken at lectures. It's kind of very, very hot area of biology at the moment. I will show you a picture. I like when this happens. It's a picture in the book that I used in the previous lecture. What chromosomal domain means is, I think it's really beautiful. Which chapter was this one? Number 5. The regulation of DNA. Could be chapter 5. Could be chapter 5. Okay, so can any of you go to the whiteboard and draw what is the structure of chromosome? If I tell you, please draw the structure of a chromosome, what are you going to write? What are you going to draw? Any of you. You can get up and draw a chromosome.
Isn't it depending what stage of the cell cycle is?
Yes. It depends on what stage the cell is at. Very much so. Because what people usually have in mind is like something like that. You know, people will draw these kind of things. You have the centromere, and then you have one chromatid, and then you have two chromatid. But this is only during cellular division, when each chromosome is going to go in the future daughter cells. Okay. Chromosomes have been duplicated, so 1 and 2, and they will be separated during cell division. But in a non-dividing cell, chromosomes are not like that. Here we can see, this is a fluorescence microscopy in which DNA has been labeled with something called DAPI or Higgs. It really depends what you are using, but it's something that makes DNA fluorescent. And when the cell is dividing, you can see that the future daughter cell are inheriting from a copy of each chromosome. And in this condition, the chromosome is really tightly packed together. Very, very, very much condensed. But in a non-dividing cell, it just looks like a soup. If you look at it in the in the normal fluorescent microscope, you can see there is DNA, but it doesn't look like there is any organization. Okay. But what you can do, if I find a picture, I will be happy. So what you can do is label all the chromosomes with a different color, because chromosomes have a slightly different sequence. So what you can do, this is possible to do that technically, is to label each chromosome with a different color. Okay, this is something that is needed to understand the next step. But then, in this case, when the cell is not dividing, and it's a normal cell who is undergoing circadian oscillations of things, like for example, hepatocytes, a liver cell, they do not divide. Very rarely they divide. So they will be happily there for many days, oscillating in rhythm to the clock. So what happens? Where are the chromosomes? Where is chromosome 1? Where is chromosome 2, 3, 4, and so on and so forth? And then we can actually check that by using a similar method as this one, but in a cell that is under normal non-dividing cell. And then you can see that chromosomes are located in very specific areas. It's not just a soup. So we have a very specific for the chromosome 9, which are purple, specific locations for different chromosomes. So there is some organization there. There is some organization. So what they are saying is that this does not show any circadian thing. It's just one picture at one time. But what they have been studying is that they have been looking, okay, is this changing with the circadian frequency? And yes, indeed, it is. There are regions in the nucleus that are enriched of genes that are transcribed highly at a specific time. Okay? So it's maybe difficult to understand, but just imagine, and we don't need this one anymore. The nucleus itself is very highly dynamic. And if I draw the nucleus here, and I use 3 colors for 3 chromosomes, you could see an example in which where each chromosome is completely random and doesn't have any organization. This is maybe the idea that most people have. I have drawn the example where there is no organization. Okay. And the gene here on the red chromosome may be actively transcribed, but the gene here in the red chromosome may not be transcribed. This could be the case. And then there is a gene here in the black chromosome that may be transcribed, and there is a gene here in the black chromosome that may be also non-transcribed. So it's completely random. But it seems it's actually not the case. It seems that somehow there are zones in the nucleus where different genes from different chromosomes currently being transcribed are located. So for example, you have chromosome here, you have another chromosome that will be here, and you have another chromosome that may be something like here. And somehow, this region is characterized by a high transcriptional activity. So these chromosomes contain very different genes, but they have organized themselves to align genes that are currently being transcribed. So this is what they say. It seems that DBP is changing its localization in the nucleus at different times. And when DBP is highly transcribed, it is in a very specific position in the nucleus. And when it is not heavily transcribed, it is in a different position in the nucleus. So we do not understand at all how this works, but it seems that this is also a component. What could be the case? Imagine what might be one advantage of doing so. One advantage of grouping genes that need to be, so let's say for example, and it might very well be the case, that, at the time where clock and BMAL bind epoxies and activate transcription of many genes, you may well find that all the genes that are activated by clock BMAL at a specific time will actually be at the same position in the nucleus. What could be an advantage of that? What could be an advantage of that?
Won't have to travel a long distance.
Yes, exactly. They will not have to travel a long distance. You can actually make sure that a certain region of the nucleus is highly concentrated in many things that are needed for transcription. Not only clock BMAL, but also polymerase II. All these proteins that are required for transcription may be highly concentrated at a specific time, and everything sort of supports each other. So that's only a possibility. We don't know if it is correct or not, but it seems to be the case. So this is an output into, so not only the molecular biology of the cell obeys the circadian clock, but also the architecture of the cell itself also seems to be under control of the clock, or it is important for the function of the circadian clock. So this is what it means, the chromosomal domain. So this is one chromosomal domain. This is a different chromosomal domain. And it seems, anyway, that the organization of chromosome in the nucleus is cyclic. This is what it is. So it's a little bit funny that it's only entering the conclusion, but they have not said anything about this earlier. But this is what happens. Okay, any question about what we have seen in this paper?
So really basic organisms have circadian rhythms.
No?
Like prokaryotic bacteria.
Yeah, that's a very good question. That's a very good question, and we are going to answer it later with the paper I brought. I like it. Nice. What other question? No? Okay, you can interrupt anytime and ask a question. Okay. So this paper was about mammalian circadian clock. And to a certain extent, it is also, you can extend similar mechanisms in eukaryotes. Okay? In flies, in reptiles, it might be very similar. There are homologs of clock genes in Drosophila. For example, cry, they have slightly different functions, but they have similar sequence. But what about less evolved organisms? What about bacteria? And for that, I invite you to look at the science report that I gave you today, the one from 2005, by a famous guy now. His English is rubbish and he gives really bad presentation. But he's called Kondo Sensei from Nagoya University. And he is very famous because he published many papers. And this one is one of the most famous papers. He is working on cyanobacteria. Cyanobacteria is a bacteria that is photosynthetic bacteria. And it was found before this paper that these bacteria also have rhythms in gene expression. Especially the expression of genes that were called Chi-A, Chi-B, and C were rhythmic. So that's about Chi-A, B, C. And there are genes. In the genome, you have Chi-A, Chi-B, and Chi-C genes. And if you measure gene expression of these Chi genes, you find they are rhythmic. So, therefore, it was proposed that the biological clock in the cyanobacteria is also like in mammals. It's a transcription translation feedback loop. Okay? So this is the first paragraph. Okay? Circadian rhythms allow organisms to coordinate their lives according to the alteration of their environments by day and by night. In most model organisms, transcription translation derived oscillatory processes based on negative regulation of clock genes are proposed as the core generator of self-sustaining circadian oscillations. So this is what I just said. Cyanobacteria are the simplest organisms that exhibit circadian rhythms. In the Cyanobacterium Synechococcus elongatus, 3 genes, Chi-A, B, and C, are essential components of the circadian clock. Negative feedback regulation of the expression of the Chi-BC operon by Chi protein was proposed as a core loop of prokaryotic TTO. In the cyanobacterial TTO model, Chi proteins do not regulate a specific set of circadian control genes, but they regulate genome-wide expression, including that of the Chi-BC operon. However, how this transcription-translation feedback loop achieves circadian periodicity and how it stabilizes circadian oscillation against alterations in temperature and metabolic activity, collectively referred to as circadian characteristics, have not been clear. These circadian characteristics are essential for the oscillator to adapt to the environment. So understanding their molecular basis is an important goal in circadian biology. So one thing maybe you may not be familiar with is operon. What does operon mean? And this is something in bacteria, not in mammals. Or at least we may find in the future that operons also exist in eukaryotes, but so far it's mainly in prokaryotes. What does an operon mean? Somebody knows?
A group of genes that are transcribed at the same time.
Yeah, exactly. So one of the most famous operon, because the first one that was discovered was that one, is the LAGZ operon. And it's an operon in bacteria that is responsible for the metabolism of sugar in bacteria. And it's in a genomic location. It's at the same location in the genome. And you have maybe a few genes, several, maybe, let's say ABC, not necessary for Chi-A, Chi-B, Chi-C, just ABC. And these genes are actually transcribed together. A long messenger RNA that contains the 3 coding sequence, and they are translated at the same time, but they make 3 different proteins. So this is called an operon. It's genes that are grouped by the fact that they are important in the same metabolic pathway, for example, the metabolism of glucose, for example, or galactose, and they are encoded by the same gene that is transcribed at the same time and translated at the same time. This is what an operon is. This is what an operon is. This does not exist, so far it has not been discovered in eukaryotes. Normally in eukaryote you have one gene that is transcribed into one pre-mRNA that is transcribed into one protein. Sometimes by alternative splicing you can have different messenger RNA, but they will be the same version of that protein. Slightly different version of that protein. This is different from the parent. Okay?
Can you have alternate expressions, or if it's only A and C are expressed?
Say that again?
Could you have, like, I guess the equivalent of alternate splicing would be alternate expressions, so only A and C would be expressed, or is it always?
Yeah, so it's slightly regulated in a different way. So you have internal ribosome antibodies where you have ribosome normally in mammals. In mammals, this is the messenger RNA, and ribosome normally they will go at the beginning of the messenger RNA. They will zip along until they meet the translational start site, ATG, and then they will start making the protein. But in bacteria, they can actually directly go inside and start without starting from the 5' end. So they can be regulated in different ways. It might be possible, not always at exactly the same ratio, 1 to 1 to 1. It may be regulated differently, but usually it's one set. Once you are committed, you actually produce the 3. Okay. So this is what is an operon. And this means what? That the chi, which one is chi B? I think chi A and B, I think. Yeah, the chi B C operon. So it means that the gene chi B and chi C, they are actually coded in the same operon. Okay. And then the second paragraph directly tells us what it's about. The phosphorylation state of chi C robustly oscillates in the cell with a 24-hour period, even under conditions where neither transcription nor translation of chi BC operon was permitted, raising doubts about the TTO model in cyanobacteria. The circadian characteristics of this oscillator suggested that the pacemaker for the cyanobacterial circadian system was not a transcription-translation feedback loop, but the chi-C phosphorylation cycle itself. Chi-C has both autophosphorylation and auto-dephosphorylation activities, and Chi-A enhances Chi-C autophosphorylation, whereas Chi-B attenuates the effect of Chi-A. These results imply that without additional kinases or phosphatases, an autonomous oscillation of Chi-C phosphorylation could be generated by cooperation between Chi-A and B. So, they say, if you take cyanobacteria and you inhibit transcription, you inhibit translation, still you will see phosphorylation rhythm of Chi-C. So this means you don't need at all transcription-translation feedback loop. So what they show in this paper very nicely, and you can directly see figure 1, what you have here is a Western blot of Chi-C. Do you understand what Western blot means? So you run protein on. Who does not understand what Western blot is? Mina wakaru? Yes, exactly. So electrophoresis from protein. You run protein on a gel. You separate protein in a gel, and then using antibodies that detect specific protein, you actually visualize the proteins you want to see. And what they have used is this technique, but they use an antibody that recognizes phosphorylated form, and the non-phosphorylated form of Chi-C. So you have two bands. In the gel that you can see in the figure, you have two bands. The bands on the top, this is Chi-C phosphorylated, and then the band at the bottom is non-phosphorylated Chi-C. This is what you have. And then they have extracted proteins from the population of cyanobacteria every, 2 hours or something. And then they have run all these samples together in the same gel, in the same membrane. And then you can see very clearly that you indeed have a very nice oscillation that persists until maybe 60 hours from the beginning. But what is amazing here is that this oscillation, it's not from protein extracted directly from cyanobacteria. What this result is, they showed similar result before, but they were extracting protein from cyanobacteria. They were killing cyanobacteria at different times. They were running this gel, they were running this Western blot, and they showed, hey, there is a rhythm in the phosphorylation of this protein. But in this case, it's not that. In this case, it's completely in vitro. What means in vitro is that they have a tube, a test tube, might be made of plastic or might be made of probably glass, it's better. And it contains some buffer, some liquid, and it contains Ka, it contains Kb, and it contains the main one. These are purified protein. They probably expressed these proteins in bacteria that are used to express proteins at very high level. And they purified these proteins. They put these 3 proteins in the tube, and then they report that simply having 3 proteins is enough to generate a very robust rhythm in Ka-C phosphorylation. What else do you need? Huh?
Phosphates.
You need phosphate, yes. But what do you need? For the phosphate, you cannot put phosphate. What do you need to put into the tube? ATP, yeah, exactly. You need ATP. And as long as you have ATP, this will keep on oscillating on and on and on and on, as long as you produce it, as long as you give ATP. And the one that is getting phosphorylated is Ka-C. It gets phosphorylated, and when it gets phosphorylated, it's a little bit more charged, because phosphates are negatively charged. It's a little bit bigger, and this is why the band is slightly on top of the non-phosphorylated one, because it's a little bit more negative, and it's a little bit heavier, so it runs a bit slower. So here they show an oscillator in vitro, and it was really the first time that anybody showed only using a few molecules, completely in vitro, devoid of any living things, they have oscillations. So that is why it was so big. They showed for the first time you do not need a transcription translation feedback loop to generate oscillations. So what they say from these observations, and it's something that they had in their mind from the beginning, is that they think that the clock in cyanobacteria, and it has been confirmed, is actually Ka-C. And via interaction with Ka-B, Ka-A, and other proteins, of course, you can imagine it's getting now very complex. There are many, many other proteins that depends on the phosphorylation of Ka-C, but this protein will then regulate cyclic gene expression of, for example, Ka-B-C transcription. So the main oscillator is this. If you kill that, you kill everything. If you kill this, Ka-C can still happily, phosphorylation of Ka-C can still happily oscillate. But of course, what will be the advantage of having this? So this is this is the pacemaker. This is the pacemaker. Please remember this word. This is the pacemaker. This is the pacemaker. And the pacemaker sort of controls rhythms of gene expression. What is the advantage of having this one? Why not only that? Why not only having a phosphorylation rhythm that affects the function of other proteins? Because this affects the function of other proteins, right? But affects transcription. But you could just completely forget about transcription. You could forget about it. And you could just say, okay, maybe by interaction with other kinases and other proteins, Chi-C could still regulate the circadian rhythms of cyanobacteria. So why do you need transcription? What is the advantage of that? Now, it just makes the system more robust. Okay. It just makes the system more robust. Because Chi-C phosphorylation has a rhythm that depends only on Chi-C phosphorylation. But if Chi-C phosphorylation regulates its own expression, that reinforces the amplitude of the clock and the robustness of the clock. Okay. So it makes the system more robust. What they show is that you only need these 3 proteins to get the clock in cyanobacteria. This is the main discovery of that paper. And then something very nice. You can check figure 1b. They also quantified the total protein amount of Chi-C, and you see it doesn't change. Of course, it doesn't change because they put the same amount of Chi-C into the tube at every time. But why did you check that? Because maybe the phosphorylation of Chi-C or the dephosphorylation of Chi-C could trigger its degradation. But it's not the case because they show that the amount of Chi-C is the same in the whole experiment. Okay, so that's figure 1. Chi-C, Chi-ABC have autophosphorylation rhythm that persists in vitro. But then figure 2 and figure 3, this is also equally important. Why? So figure 2 shows that, well, they are testing the phosphorylation rhythm at different temperature. This is figure 2. And they are checking some mutants of Chi-C and Chi-C wild type and mutant strain, and they check the period. Why it is important? Why do they do this? You can read the paper, and then if you find an answer. First of all, figure 2. What is the importance of that? Why do they do the experiment in figure 2? Why are they checking the impact of temperature?
The characteristic of the circadian rhythm is that it doesn't impact any temperature.
Yes, exactly. So in the very beginning of this lecture, I told you that there are mainly three essential characteristics of the clock. One, as you said, is its temperature compensating. It should not be too much affected by temperature. What is the first one? What are the 3 characteristics of the clock?
It can repeat by itself.
Yeah, it is endogenous. Okay, it is self-sustained and endogenous. it is temperature compensated, and then
It's 24 hours.
it is.
It's 24 hours. It's 24 hours.
That's also, it's not, yeah, there are clocks of 2 hours, and there are clocks of 1 year, but they are still clocks. 24 hours is for circadian clock. What is the third important characteristics for a biological clock? More for its importance for the organism. Yes. It must be entrained by the environment. The clock is not only to have a self-sustained clock in your body, but it's to be able to be in phase with the environment and anticipate changes in the environment. This is really the main thing. So the figure number 2 is to show, okay, we say that chi ABC is the basic circadian clock of cyanobacteria. So if this is so, then it should be temperature compensated. So if we change temperature, we should expect that the rhythm of Chi-C autophosphorylation, dephosphorylation will not change much. Why? Why it's important? Why it's important? Okay, you can see that the graph they have made. So they checked 25 degree, 30 degree, and 25, 25, 30, and 35. And there are some differences. You can see that the temperature has an effect on the clock. So the hotter it gets, the smaller the period is. The shorter the period is. This is figure 2b. It's about 22 hours at 25 degrees, but it's about maybe 21 hours at 35 degrees. So it is temperature dependent, but it's not as much as you would expect from enzymatic reaction. What would you expect from a reaction that is not temperature compensated? What would you expect? So you have a graph here. You have 25, you have 30, and you have 35. And you have maybe 22 hours period. And at 25 degrees, you have a 22 hours period. What would you expect if it is not a temperature compensated reaction?
The period would decrease as the enzyme activity is faster if the enzyme denatures.
Faster, like about what?
The rate of it?
Huh?
The rate of it?
Yeah.
Decrease?
So the period will
Decrease.
decrease much more, right? Maybe. And what is the measure of that? How do you measure? What is the biochemical measure of that? What parameters are we talking about here? Do you remember the name of the parameter we are talking about? The temperature compensation, yes, but what does it mean? How do you name this temperature dependency of enzymatic reaction? It's the Q10, right? It's the Q10. It's the Q10. So for every change of 10 degrees, you should have a change in enzymatic activity. And typically, for example, for muscle contraction, the Q10 is about 2, which means for every change of 10 degrees, the enzymatic reaction goes twice as fast. The clock has a Q10 of 1. So it doesn't change. Theoretically, it doesn't change. What does it mean, a Q10 of 2 for the clock?
be 11?
Yes. It should be 11, twice as fast. It should become 11. But it's not. It's not. It's barely maybe 1 hour. So the first point is made. This endogenous, this self-sustained phosphorylation rhythm of Chi-ABC complex is temperature compensating. Then they are checking whether mutations that have been made in cyanobacteria and that have been shown to have an effect on the period of the cyanobacteria behavior and metabolism, they also here have an effect on the period of these phosphorylation rhythm. So this is very strong data. This is extremely strong data. Here we have a clock that only depends on phosphorylation. Okay, so you are a scientist, and you are thrilled and motivated by all things biological, and you read this. You read this paper. What are you going to do? You are studying. Your passion is not little bacteria. You like mice. You like whatever else. You like flies. You don't study bacteria. You read this. What are you going to do?
Something similar in other animals.
Yeah, absolutely. All right, so we have phosphorylation rhythm here. Actually, there might be phosphorylation rhythms in mammals. There might be phosphorylation rhythm in eukaryotes. So, what I am telling you now, take it with a pinch of salt. It means it might be completely incorrect. But let's just imagine, what kind of experiment would you do to show that or to investigate? We don't know. Currently, no phosphorylation rhythm has been found. Okay. People are looking for it, but they have not found it. But what is one thing that you could do to check whether there is a phosphorylation rhythm in mice? What could you do? I have not done it. I kind of want to do it, but I have not done it because I don't have the capacity necessary in the laboratory here. I may do it in the future. What could you do? What could you do? And the hint is maybe think about what I'm telling you. I'm not sure this is true or not. It's only possibilities. If you want to investigate phosphorylation rhythms in mammals, what would you do? Think about evolution. Cyanobacteria and us is quite billions of years apart. But what could you do? What could you do?
Check how conserved the genes are.
Check up?
How conserved the genes are.
Yeah, that's a very good start. Be more precise.
So you see what the sequences of the genes for the Chi-C in cyanobacteria.
Yeah, exactly. So you can look in the mouse genome whether there are any proteins who have a sequence similar to Chi-C. Yeah. And if you actually do that, you find. you find some protein that have a very close sequence to Chi-C. And Chi-C is part of a family of kinases that is called AAA, triple A, family of kinases. And you find proteins in mice that are very similar to Chi-C. Maybe sequence is quite far apart, but they are part of the same family. And there is a very interesting thing, is that the Chi-C, maybe I can show you, I have slides for that. The Chi-C has a very, very interesting structure. It does not operate as a monomer. It does not operate as a monomer. But I think I have chronobio. You see, I had this lecture plan for that's which is that the one? Okay, yeah, so I can show you this one to show that it became a little bit more complex that I showed you before. So that's in the slides I put on classes before at the beginning of the lecture, but that's currently simplified model of how the clock in cyanobacteria works. So this is the Chi-A, Chi-C, Chi-A, and Chi-B here. But they are other, you don't need to remember all the proteins, okay? Just remember how this works. So Chi-A, Chi-C, Chi-B, they are of course encoded by their own gene. There is another protein called SAS-A here. And there is Sig-A, which is also important to feed into the oscillator the effect of temperature or the effect of light for entrainment of the clock. But essentially the Chi-C is really the protein that actually phosphorylates itself, and then via the phosphorylation of Chi-C and the regulation of SAS-A, which regulate by phosphorylation a transcription factor that regulates different class of genes with different phases, just like in mammals. Different phases regulated by different things. So this is actually how more or less it works. But the main oscillator is really the Chi-C, Chi-A, Chi-B, Chi-C family. And so this is what you just saw now, but I want to make this little guy bigger because the structure of this stops every time. The structure of Chi-C is very interesting. You can see Chi-C is actually a hexamer. And each Chi-C subunit can be phosphorylated. So it's kind of a ring. And the phosphorylation is sequential. It's really like a cycle of each subunit is phosphorylated at its turn. So there is a lot of work that has been done there at exactly what is the sequence of phosphorylation. So it's very, very complex. But very interestingly, there are proteins in mice that have exactly the same hexameric structure, that are part of the same family, that have exactly the same structure. But whether they can undergo autophosphorylation cycle is completely unknown. But interestingly, these proteins in mice that have the same structure, they are mitochondrial protein. Why is that interesting? Why is the fact that a protein that is one of the closest proteins to Chi-C is a mitochondrial protein. Why it is kind of exciting? I think, for me. Why do you think it's exciting? Why is it exciting? Why is it exciting? Yeah, for example, that's one of them here. That's one of them here. That's one of them here. It's a hexamer, so it has partners, but it's a hexamer, and it's a mitochondrial protein. But why it is exciting that it's a mitochondrial protein? Yeah, that's also interesting, but that's not what I was thinking about. Why do you think? What is the origin of mitochondria?
Bacteria.
Yeah, it is believed that the origin of mitochondria are bacteria. So I find it's kind of a little bit interesting because it might be possible that if it's a protein that goes into the mitochondria and influences mitochondria metabolism, we may well find that actually some part of the mitochondria metabolism or something is controlled by the clock via a phosphorylation cycle of these proteins. So this is only a possibility. But anyway, yeah. So the main question is, do we have transcription, translation, independent rhythms in mammals? And people have looked at that. Scientists have looked at that, but most were not successful. And there are actually two sides in circadian biology. There is one side that firmly believes that the circadian clock in mammals is not transcription, translation, feedback loop mediated. Despite all the evidence, they firmly believe, nah, it has to be phosphorylation, it has to be protein mediated. And there is another clan which firmly believes that it's actually mediated by the transcription, translation, feedback loop. And of course, this clan is the one who actually discovered the clock genes. Okay? So it's no surprise that there are two clans that oppose each other because they try to preach their own discoveries. But, very interestingly, and that's the second paper I gave you today, circadian clocks in human red blood cells. This is by a young guy called John O'Neill. It's in 2011. And he has published in Nature 3 papers on exactly the same subject. This, together with another paper showing that in algae, he also found the same rhythm. And the year after, in 2012, he again showed the same thing, that in many organisms, he found the same thing. Without transcription in eukaryotes, you can still see rhythm, not in phosphorylation, but in redox. In oxidation reduction, of a specific protein called peroxyredoxin. And you can take a look directly if you want in the picture figure 1a. And this is similar to what you have seen before in the Chi-C. It's a little bit less clear maybe, but they have measured the amount of peroxyredoxin oxidation. So this is hyperoxidized peroxyredoxin with the circadian time. So you can see it's low at 0 and low at 24, but it's high at 12 and 36. So why it is related to the same subject? Why showing a rhythm in peroxyredoxin oxidation, why is it linked to the previous thing that I showed you?
Circadian is not created by transcription.
Yes, of course. So peroxyredoxin oxidation, it's not caused by transcription translation. But how can they say that? Because I could say, all right, so I think there is a protein that oxidizes or reduces peroxyredoxin that is responsible for this oxidation. And this protein may be regulated by the clock via transcription translation. What is very specific about red blood cells? They don't
Have the nucleus.
They don't have nucleus. They don't have DNA. They don't have DNA, so they don't have transcription feedback loop because they lack DNA. Moreover, these experiments were not made in vivo. They took red blood cells from human patients and they cultivated the red blood cell in a dish at specific temperature, and every 4 hours or so they checked what was the oxidation level of peroxyredoxin. And then they show that peroxyredoxin rhythm have a rhythm. And indeed, they confirm, just to be sure of what they are saying, so this is figure 2, they are using inhibitors of transcription and inhibitors of translation to show that even with inhibitors, the oscillations in peroxyredoxin oxidation remain. And then, very similar to this paper in cyanobacteria, in the figure 3, they show that these rhythms are temperature compensated. So they tested 32 or 37 degree and sampled every 4 hours. But as you see, the oscillations are very, very similar between the two samples. Maybe the level of the protein changes, but the oscillations are very similar. So every time they show a picture, there is loading control. Do you see? Under the peroxyredoxin SO23. So this is the hyperoxidized form of peroxyredoxin protein. So they show the dimer, and then underneath they have the loading control. What is the purpose of the loading control? Do you understand this? This is another protein. I don't remember what they use as a loading control, but people usually use proteins that will not change at all, like a cytoskeleton protein or something like this. Why do they show loading control?
Sure that that's not rhythmic.
Yeah, so with the loading control, they actually tell us that even though we can see more or less a rhythm in peroxyredoxine superoxidation, it is not because we have loaded different amount of protein. So this loading control makes us able to say, okay, at every time they loaded the same amount of protein. So the rhythm we are looking at, it's really a rhythm that is specific of peroxyredoxine oxidation. Okay, and that's where it is at. We don't know what generates this rhythm. And it has not been confirmed either by any other team that they own one. Some scientists in the United States, this is a guy from Cambridge, many scientists in the United States have tried to confirm these data, but they did not manage. They did not manage. Other scientists could not show rhythm in peroxyredoxine oscillation. Using the protocol explained here, they could not detect any rhythm. So whether this is true or not, it's not clear yet. It's not clear. The papers are well accepted and they have not been retracted like the STAT paper or whatever, but some people do not really believe that this is really happening. Okay. Because unlike the transcription translation feedback loop, that 100 teams have shown the same thing, this has only been shown by one team in Cambridge. Multiple times, but only by themselves. So, whether it's true or not, we don't know. But we have to presume it's true, because it is published, and they have published many papers on the subject. So we have to say, okay, so it seems to be true. But how it is generated, we have no idea. Unlike the Chi-C story, we know Chi-C are generating their own autophosphorylation rhythm. In this case, what generates oxidation rhythm, we don't know. Because in this case, it's not peroxyredoxine in a tube. It's peroxyredoxine extracted from red blood cells. So we don't know the mechanisms. The jury is still out as to whether there is really a clock in human cells that does not depend on transcription translation feedback. And if there is, as this suggests, what it is, it's not. Okay, and I think that's the end for today.
In the Chi-C story, is there any kind of concentration of is there a kind of a ratio of phosphorylated Chi-C in that if we start at that ratio, then the oscillation doesn't happen? For example.
That's a very interesting question. So you're wondering whether if you start, say, at a completely unphosphorylated Chi-C.
No, at the ratio in which the two get in equilibrium. So I don't know. It is.
See, that's interesting. So you imagine a perfect equilibrium between phosphorylated and unphosphorylated Chi-C, that they just stay constant.
So if we start at that ratio, then the oscillation occurs.
That's interesting. I don't think it is. I don't think this has been shown. but it might be possible. But I think there is a mechanism to prevent that from happening. Because of the Chi-C protein being made of 6 subunits with a sequential phosphorylation, I think in this paper, they didn't know what is really the phosphorylation cycle. But maybe next time I will bring some data to show that this kind of equilibrium cannot happen because of how Chi-C function. So, but it's kind of a good idea. It's kind of a good idea. Maybe there is, and it has not been shown yet. And it's kind of interesting because in one of the latest papers in Science from the same team, you can imagine, we are looking at a phosphorylation cycle that takes 24 hours. Phosphorylation is very fast. Phosphorylation is like maybe 1 second, 1 millisecond. And dephosphorylation is as fast. So how come we have a phosphorylation-dephosphorylation cycle that lasts 24 hours? And they show very nicely in one of their latest papers is that the shape of the Chi-C protein is actually promoting very inefficient phosphorylation. Some kinase, they will phosphorylate very quickly. But Chi-C actually inhibits phosphorylation. The shape is not, it's not optimal for quick phosphorylation. The shape of Chi-C is optimal for very slow phosphorylation. So that one complete cycle is 24 hours. Usually kinase, they are selected for their high efficiency, but Chi-C has been selected for very low efficiency in terms of the phosphorylation reaction. But of course, its efficiency as a circadian oscillator is very high. Okay, you can leave whenever you want.